Hello, my name is Jacqueline Penaloza, and I'm a PhD candidate from Ohio State University. I am from Peter White's lab, and today I will be presenting on Multiclass Classifier for predicting congenital heart disease subgroups using data from copy number variants. Congenital heart disease is a defect in the heart present at birth, which can impact 1% of live births worldwide. The genetics of congenital heart disease is not well understood. One possibility is copy number variants, also known as CNVs. These are structural gains or losses in the chromosome, and unfortunately, this can impact many genes. The motivation behind the study is that often congenital heart disease patients who have the same malformation are diagnosed with different copy number variants, and how congenital heart disease forms from this variant is unclear. Therefore, the goal of this study is to functionally annotate copy number variants linked to congenital heart disease by using machine learning. In this study, we leveraged our access to data from the Cytogenomics of Cardiovascular Malformation Consortium, also known as CCVM. This dataset includes information on copy number variants and clinical notes about the patient's diagnosis. For the machine learning, we used patients who had isolated congenital heart disease, and we only focused on copy number variants that have impacted protein coding genes. The type of CNVs that we focused on are either gains or losses. In order to remove functions that might not be particular to congenital heart disease, we used control data from DGV dataset, also known as the database of genomic variants, and we focused on CNVs that are greater than 5 kilobases long. The remaining functional annotations we then inputted into our machine learning along with clinical features. As illustrated in the graph below, you can see that the number of copy number variants in each congenital heart disease subgroup is imbalanced. In order to overcome this issue and optimize our conditions for machine learning, we applied oversampling by SMOTE, also known as synthetic minority oversampling technique. This method creates new synthetic samples in order to balance the copy number variants in each group. You might be wondering, what is the difference between a multi-class classifier and a binary classifier? A binary classifier is when you're trying to predict two outcomes. In this example, we're predicting a healthy patient versus a diseased patient. By random chance, our baseline is at 50%, or simply considered like a coin toss. And for a multi-class classifier, we have more than two outcomes. In this example, we're trying to predict 8 different diseases, and our baseline lowers from 50% for binary to 12.5% for this 8-way example. We evaluated tree-based algorithms for predicting congenital heart disease subgroups. We evaluated random forest, XGBoost, and decision tree. If we recall what we learned from the last slide, the baseline for an 8-way classification is 0.125. Therefore, random forest performed 5 times greater than the baseline across all metrics and was selected as the final model. The confusion matrix on the right is for random forest. On the y-axis, we have the true label, and on the x-axis, we have the predicted label. We used Shapley additive explanation, also known as SHAP plots, to analyze the features that most contribute to the classification of congenital heart disease subgroups. On the left, we can see the global SHAP plot, and on the right, we can see the class-specific SHAP plot. Overall, the global SHAP plot on the left revealed that the most important parameters are the type of CNV and generally the brain-related pathways. On the right, the CNVs that appear to be important for heterotaxy tend to be involved in CNV duplication and impact to cell adhesion and projection with distinguishing features in tissue morphogenesis and positive regulation of intracellular signaling transduction. In contrast, on the right, we have the LVOT subgroup. We see larger weight placed on the central nervous system development and neurogenesis pathway. In conclusion, we found unique and shared features among each congenital heart disease subgroups. Generally, our results indicate that machine learning is an effective tool for functionally annotated copy number variants linked to CHD. For future direction, currently our model only focuses on protein coding genes that impact CNVs. To expand this, we hope to include information that will help us annotate non-coding genes as well. The clinical application of our machine learning tool is that during the first trimester of pregnancy is when chromosomal abnormalities are screened. CNVs can be assessed with our machine learning method to diagnose these congenital heart disease and to understand their function as well. Although echoes can detect congenital heart disease and are very accurate, echoes cannot interpret copy number variants. Therefore, our model, alongside with echoes, can increase the diagnosis and outcomes for patients with congenital heart disease. I would like to thank you for listening to my presentation, and feel free to post your questions on the website.